important for neuromodulation when you think about it. It's pretty important for neural transduction. You wouldn't want the neurotransmitter to just be out there and hanging out until it diffuses away. You want these actions to be very precise, and you want neurons to be able to communicate to their postsynaptic neurons, but in a very concise manner. Okay? And so you can actually change synaptic strength by a number of ways. One, you can increase the number of neurotransmitter receptors that you have, right? You're going to get more of a response if you have more receptors on the postsynaptic neuron. Two, you could change the rate at which the neurotransmitter is taken back up into the axon terminal of the presynaptic neuron, right? And third, you could change the rate at which you synthesize neurotransmitter from the get-go. So a lot of pharmaceuticals that you take that are neuromodulators, they target any one of these particular types of targets, essentially, these mechanisms. You have drugs that will target the neurotransmitter receptors. You have drugs that will actually cause a degradation of the neurotransmitter in the synaptic cleft. You'll also have drugs that will actually block the breakdown of the neurotransmitter in the synaptic cleft. You have drugs that will cause inhibition of reuptake. So if you don't have neurotransmitter being taken back up into the presynaptic neuron, you're going to have more of the neurotransmitter out in the cleft, and that's going to cause more signal on the postsynaptic neuron. So different pharmaceuticals, and we'll talk about various ones, but different pharmaceutical drugs that we usually think of as, pharmaceuticals that deal with psychokinetics are targeting the synapse. Okay. All right. So let's go back for a second and talk about calcium as the signal here. So before that action potential is sent down the axon, these particular vesicles are tethered to the membrane. So the neurotransmitter has been synthesized via normal protein synthesis in the cell. Those proteins, they're typically proteins, have been packaged into these vesicles, and the vesicles are sitting there anchored at the plasma membrane waiting for that calcium signal. So there are two proteins that we want to talk about that are serving this anchoring function, and those are called the snares. So there are vesicular snare proteins, the ones shown in blue, that are targeting or that are sitting on the vesicle membrane. And then there are membrane snares, called the T-snares. And I had to write it down because snare is such a long name. Snare stands for N-ethylmaleimide sensitive fusion proteins. No, that's okay. Snare. Snare. Okay? So vesicular, certain things like inositol triphosphate and phosphoinositol diphosphate, things that kind of roll off the tongue. This is, you just know snare, okay? So these are specific proteins that will tether. So what you can see here is that the plasma membrane has T-snares, and the vesicles have V-snares, and there's sort of a lock and key docking. We call it docking. And these vesicles will dock with the vesicles that have the V-snare proteins will dock with the associated T-snare proteins. Here are your calcium channels that are voltage gated, sitting on that axon terminal. And once you get the depolarization event happening from the action potential, the calcium channels will open, and calcium is the signal that will cause a conformational change, allosteric modification, right? That will cause a protein structural change between the V-snare and the T-snare, and that will cause an undocking and a fusion event of the vesicle with the plasma membrane. This will cause the neurotransmitter, which is shown here in the little black circles. The neurotransmitter can then be released into the synapse, and here is that waiting postsynaptic neuron that's got the neurotransmitter receptors that will be bound by that particular neurotransmitter. So, what you guys should be able to do now that you know all the steps of neurotransmission is you should be able to start from the cell body of a neuron, right? You should be able to tell me about the graded potentials that happen in the cell body, right? The EPSPs and the IPSPs that are downstream of specific neurotransmitter receptors. I'm going to say a few more things about those as we talk about the classifications of neurotransmitters. You should then be able to tell me a little bit about summation of those graded signals and that summation is going to, in order for an action potential to be propagated down the axon, you're going to need to pass threshold, right? That it's going to have to be a depolarization signal that's strong enough to cause those voltage-gated sodium channels to fire, right? Then you need to know the steps of an action potential and about action potential propagation and conduction velocity, the importance of myelin and the importance of axons that are large in diameter. And then once you get to the axon terminal, you should know that that action potential depolarization wave will cause these voltage-gated calcium channels to open. Calcium will rush in. It will activate changes in V snares and T snares such that these vesicles will fuse with the plasma membrane, causing exocytosis of that specific neurotransmitter, right? So as you're studying for this, remember the Hinkle scratch paper method? As you're studying for this, take out a stack of scratch paper and just start writing these steps over and over and over again. Go back to your notes. If you missed a step, do it again. And then if you missed another step, do it again. And just, if you sit there for probably 20 minutes and write all the steps of neurotransmission down and the important, and remember that I'm a mechanism person. I really want you to know why these things happen, not just a rough overview of why these things happen, but specifically what proteins are involved in each of these steps. You guys know that by now, right? You know that you need to know, like, that the sodium and potassium voltage-gated channels are responsible for the action potential, etc. And so once you commit that to memory, this will be something that you can regurgitate on an exam, right? Okay, so let's talk a little bit about neurotransmitters and neuromodulators. They're called neurotransmitters, and there's another class called neuromodulators. The difference here is that neurotransmitters are ones that will cause an acute response, like an opening of a channel, or they'll cause an electrical potential in the cell body. A neuromodulator is actually going to cause changes in gene expression. So I could be a neurotransmitter that activates a receptor, and instead of leading to another action potential or leading to an EPSP or IPSP, maybe I cause this neurotransmitter to make more maybe I cause this neuron to make more receptors. So in that way, you can modulate, you can be a neuromodulator that actually has a more long-term effect on the neuron and how it will behave. I don't know if some of you have heard of synaptic strength or long-term potentiation. I know some of you psych majors maybe talk about long-term potentiation. That's basically learning. So long-term potentiation, which is called LTP, that's the acronym, that is thought to be one way by which neuromodulation happens. So when you think about a task, for example, last week when we went over the G protein coupled receptor pathways, right? In the beginning, you didn't really know the pathway, and you sat down and you sketched out the pathways, and you hopefully sketched them over and over and over. Well, you obviously know that in some time you can recount those details much better than you were able to before, right? And that's learning. So in very basic terms, that's learning. But these are actually modulations that are happening in your neurons. You're stimulating learning via changes in gene expression. So what does that mean? Well, you're actually causing synaptic strength. You are making better connections between your neurons, right? And so how might you be able to do that? So let's just say that neuron 1, the presynaptic neuron, and neuron 2 are very important to do some tasks, like regurgitate G protein coupled receptor pathway or something, okay? And before, this synapse wasn't used very much before. It was there, but it wasn't really you weren't training it, right? You weren't really using it that much. So as a response, the cell isn't going to do much work to make you a whole bunch of neurotransmitter receptors if you're not having a whole lot of neurotransmitter around, right? It's sort of a feedback response. Like, I'm not getting a whole lot from this presynaptic neuron, so why would I spend the time making all these neurotransmitter receptors? So in that sense, the synapse would be weak, right? Because it's not going to, you might get some neurotransmitter, but you're going to have very few receptors, so you're going to get a small response. However, if you start sending action potentials down this presynaptic neuron more and more and more, you're going to get more neurotransmitter released into that synaptic cleft. And if you're a neuromodulator, you might activate this neuron to actually start making more neurotransmitter receptors. Whoa, suddenly we have more of this neurotransmitter, or more of this neuromodulator, this synapse needs to, it's just like training a muscle. This synapse needs more. This synapse needs more receptors. This synapse needs more machinery in order to communicate these signals. So this is what's happening in learning and long-term potentiation. These chemicals are actually being released more often because you're using these neurons more often. And those neurotransmitters or neuromodulators can totally change the features of the synapse and either increase the strength of that synapse, or if you don't use it, then you can decrease the strength. That, Tim?
Is this only for long-term memory or short-term memory?
It's for both. It's in the simplest terms, any neuronal change is going to be responding to neurotransmitters and the frequency with which you fire these neurons, right? And the concentration of neurotransmitter that's there. It gets very complicated, right? Because like we said this morning, you can have, I could have hundreds of neurons all synapsing on one postsynaptic neuron, and they all might be releasing a various milieu of neurotransmitters, right? Each one would be releasing one kind, but now this guy has to respond, and imagine the combinations you could come up with. On day one, if there's half of those neurons that are firing and you're getting 50% of those neurotransmitters at 50% concentration, that might cause one type of neuromodulation, versus if you had all of them firing all the time, summating those signals, right? So it can get very complicated, but that's exactly how we cause synaptic change. So long-term potentiation is thought to be just a more obviously long-term versus short-term. These are changes that are going to be the result of gene expression changes. So you're going to cause the cell to now start making more neurotransmitter receptors, or making more snare proteins, or making more, the cell itself will make more neurotransmitter and load more of it into the vesicles. So again, think about how synapses could be modified, right? If you have to have voltage-gated calcium channels to cause calcium to come in, if I increase the number of calcium channels, wouldn't that maybe cause me to release more neurotransmitter? Yep. If I increase the concentration of these snares, or T snares, if I increased how much neurotransmitter is actually made in this neuron, then I could load more at the end, and that neuron could release more neurotransmitter. If I increase the number of receptors on the postsynaptic neuron. So I say this because our neural communication is extremely complex. If you think about the combinations of neurotransmitters and also the different proteins that could change in their own synthesis and concentration, can obviously vastly modify how these neurons communicate with one another. I want to add one more level of complication, and that's in the synapse, so extracellularly, there are enzymes that serve to break down neurotransmitters. So let's just take an example. Acetylcholine, you guys now know that acetylcholine is used throughout the sympathetic nervous system, and it's also the neurotransmitter that's involved in stimulating skeletal muscle. So acetylcholine is a hugely important neurotransmitter in our peripheral nervous system. So if this was a cholinergic neuron, remember the tip off there is choline, so acetylcholine. If I'm a cholinergic neuron, then I release acetylcholine. And on the postsynaptic neuron, I would either need a nicotinic or a muscarinic acetylcholine receptor, right? The nicotinic is a cation channel that opens up a conductance to sodium and potassium, right? The muscarinic actually is a G protein coupled receptor that's coupled to GQ. So either one, I need an acetylcholine receptor on the postsynaptic membrane. We'll call that an AChR. So if I were to say I want to increase that strength of that synapse, maybe this particular connection is going to make me learn my physiology a little better, right? So make sure that you understand which ways you could increase the strength, right? I could make more acetylcholine, right? I could make more calcium ion channels. I could make more B snares and T snares so that I could assemble more vesicles at the axon terminal. I could make more acetylcholine receptors. So now I've said those a few times. But there's one more detail, and that's in the synapse there are enzymes that will break these neurotransmitters down. And acetylcholine specifically is broken down by cholinesterase. It's called acetylcholinesterase. And I just abbreviated ACH esterase. And what that does is it takes acetylcholine and it breaks it apart. And it's going to break it into choline and basically an acetyl group. So it's going to just cleave it down to choline. So choline can't bind to the nicotinic or muscarinic receptors. So once acetylcholinesterase has had its time to work on acetylcholine, that neurotransmitter is essentially gone, right? Even though it didn't have to be taken back up into the presynaptic neuron, it's no longer active. So if you're following me, again, acetylcholinesterase is an enzyme that's always hanging out in the synapse. It's always there. So you get acetylcholine dumped out into the synapse. Some of the acetylcholine is going to diffuse across the synaptic cleft and bind to the receptors, but some of it's immediately going to get broken down, right? So isn't it true that if I actually blocked acetylcholinesterase, that I could increase the amount of acetylcholine that could bind to the postsynaptic neuron? Have you guys ever heard of sarin gas? Sarin gas is a neurotoxin. It's a nerve agent. It's a nerve gas that's used in biowarfare. And the most famous example of where sarin gas was used was on a Japanese bullet train, probably now 10 years ago. And someone released sarin gas in the train, and it killed a bunch of people. And sarin gas is an acetylcholinesterase blocker. So if sarin gas blocks this, and what happens is you might think, well, why would too much acetylcholine be bad for the postsynaptic neuron or the postsynaptic cell? And it turns out that the acetylcholine synapse is also used for skeletal muscle. So you could superimpose this cell as muscle, okay? And we'll talk a lot about skeletal muscle physiology, so just imagine for a moment that this isn't a neuron, but this is a muscle. Same thing happens. The motor neuron in this case sends a signal down the axon, calcium rushes in, the V snares and T snares change shape and cause an exocytosis of acetylcholine into the cleft. Acetylcholine binds to in this case, the nicotinic acetylcholine receptor. That causes sodium direction, and you get a muscle action potential that happens. Well, yes, so what happens is having a very small appropriate amount of acetylcholine turns out to be just as important as having any at all. And what happens is if you block acetylcholinesterase with sarin gas, you have too much acetylcholine in the cleft. And too much of a good thing is not good, right? So what happens is that you have too much acetylcholine. This causes a long-term depolarization. And can someone remind me why that might be bad? Yes?
Permanent muscle contraction almost?
Well, that might happen at first, but what do you have to do to the membrane in order for inactivated sodium channels to reopen?
You have to repolarize it?
Yeah, great. Do you guys remember that? That I'm the sodium voltage-gated ion channel, I get depolarized, right? It opens up, sodium rushes in, but the inactivation gate gets stuck in there right away. And the only way to get that inactivation gate out of there is to repolarize the membrane. But sarin gas is making a situation where I'm not repolarizing. So it doesn't matter how much more acetylcholine is in the cleft. If you don't repolarize the membrane, can you have another action potential? Not in a neuron and not in an electrogenic membrane like muscle. And so people die from sarin gas and other nerve agents like this because of paralysis. At first you think, yeah, it's too much, so you have like constant contraction. But it's actually the opposite because it's all about electricity. Because you have this constant depolarization, those sodium channels that opened once and then got plugged up, they're just still plugged up. There's no way you can get another action potential. So you can't contract your skeletal muscles anymore. And well, that's okay, maybe I'm on the train and I'm sitting there going like this and I can't move. But why would you die? You can't breathe. Your diaphragm muscle is a skeletal muscle, right? And the skeletal muscle that controls your lung movement will not be able to contract either because it's completely controlled by acetylcholine-releasing neurons. And so that's how people die from sarin gas. When we get to muscle, we'll talk about a couple other agents, including botulinum toxin. You guys have probably heard of botulism, right? Botulinum toxin. And there are some other nerve agents that, carbon monoxide, other nerve agents that will cause paralysis. So the end result is usually the same, but the mechanism of the drug is different. Okay, so let's look at the classes of neurotransmitters and neuromodulators. In your book, there are 6 classes, but I just lumped the last 2 classes into one because they're sort of others. But anyway, let's give you some details about these. Acetylcholine, you might want to have in your notes, is extremely important in your peripheral nervous system. It pretty much dominates the peripheral nervous system. And I'm going to fly back to one of our very first slides here and remind you of this particular slide. So remember, what you're looking at are the divisions of the nervous system. And you can see that acetylcholine is, so here's your central nervous system, and all these neurons are coming back out into the periphery. And look at how common acetylcholine is used. First of all, your somatic nervous system, which is controlling your muscle movement, right? Your skeletal muscle, your voluntary activities. The only neurotransmitter that your motor neurons release is acetylcholine. The only way that you can stimulate your skeletal muscles to contract is via acetylcholine release, right? The only other way is through an e-stim machine, right? That bypasses acetylcholine. But to get that muscle action potential that's required, you need to have acetylcholine bind its receptors. Here it is again. So here's a parasympathetic neuron. Remember that that's part of the autonomic nervous system. Do parasympathetic neurons mediate fight or flight or more housekeeping functions? Housekeeping, like lowering your blood pressure, lowering your heart rate, digestion, secretion of hormones, right? Things that are happening in you right now, right? Digesting your food and feeling sleepy time, right? That's parasympathetic neurons. And the acetylcholine is released both by the presynaptic and by the postsynaptic neuron, postganglionic neuron. Even in your sympathetic neurons, so your fight or flight, the ones that mediate fight or flight, the presynaptic neuron, the one that is going to synapse here at the ganglion, where this synapse is in the peripheral nervous system, the preganglionic neuron releases acetylcholine. The postganglionic releases the one you're thinking of, which is epinephrine, right? Norepinephrine, which is adrenaline. So you're thinking your fight or flight. Well, I thought that was adrenaline. It is, but the first neuron in the chain releases acetylcholine on the second, and then the second releases epinephrine. So again, the acetylcholine is incredibly important in the peripheral nervous system, both for somatic, for parasympathetic, and for sympathetic, right? All right, now we're going back on our little neurotransmitter, neuromodulator hunt. So acetylcholine is very important for the peripheral nervous system. Another class of neurotransmitters, neuromodulators, are called the biogenic amines. This includes dopamine, norepinephrine, and epinephrine. Those are the catecholamines. This group also includes serotonin and histamine, which you guys probably know from, like, if you have hay fever, right? So again, all of these are neurotransmitters or neuromodulators that will be released by specific neurons. Many of these are important both in the central nervous system and in the peripheral nervous system. You have another another class of neurotransmitters and neuromodulators that are derivatives of amino acids. So you take your singular amino acids, you modify them a little bit, and they actually themselves are neurotransmitters. Glutamate, which is the most abundant neurotransmitter in the central nervous system. So in your notes, you add to this that that's the most abundant neurotransmitter in the central nervous system. And here's another detail. So 50% of neurons in your central nervous system, approximately, are glutaminergic. They release glutamate. And I'll give you more on kind of the mechanism of action. By the way, all of these that I've talked about thus far, they evoke excitatory postsynaptic potentials. They open up ion channels that are going to cause depolarizations. So all of these, acetylcholine, the catecholamines, serotonin, histamine, they all will cause EPSPs. They're excitatory neurotransmitters. Glutamate is also an excitatory neurotransmitter. And I'll talk about the receptors they act through in a moment. The first one that's inhibitory, and the one I'm going to focus most on, is the one that we talked about this morning, which is GABA. That's gamma aminobutyric acid, and GABA is an inhibitory neurotransmitter that is going to stimulate chloride channels to open. So if you had a lot of GABA around, is the postsynaptic neuron going to be firing more or less? So everybody, it's going to be firing more or less. So again, let me say it again. If I have more GABA in a synapse, is the postsynaptic neuron going to tend to fire more or less?
More.
The less is habit, right? Because it's an inhibitory neurotransmitter, right? And so, would you maybe, if you were a pharmacist or a person who worked in a pharmaceutical company, and you wanted to sort of inhibit some sort of neuronal pathway, wouldn't that be one way you could do it? Is to maybe make a drug that acts like GABA? So has anyone ever taken Valium? So Valium, you guys all kind of know what it does though, right? We usually know it as something that you take before, like if you have a fear of flying, the physician will give you some Valium, right? It's an anti-anxiety medication, and it sort of will put you in a little bit of a dream-like state, right? It's sort of like, it has an inhibitory effect on the central nervous system. Valium is a GABA agonist. It's a GABA receptor agonist. That means that it acts like GABA. And so that's its mechanism of action, is that instead of having lots of GABA around, you put a bunch of Valium in your bloodstream, and it stimulates those receptors and causes your central nervous system to be more on an inhibitory plane, right? The other thing that will bind to GABA receptors is alcohol. So alcohol, as you know, has a depressive effect, and alcohol will bind to GABA receptors and cause that same cognitive inhibition, right? Incidentally, ethanol also binds to glutamate receptors, which themselves are excitatory. So it sort of has this kind of bipolar action, but overall, ethanol is a neurodepressant, right? It's a neuroinhibitor. And the inhibition comes from it binding to GABA receptors. So more chloride will come into the cell. You'll get less of a chance that you're going to elicit action potentials in those downstream neurons, and you'll sort of be in more of a, that's why cognitively, I know none of you ever are in that situation, but cognitively, if you drink a lot of alcohol, you have a lot of trouble with slurring your words, etc., right? And that's because of the GABAergic effect that it's having. So both Valium and ethanol can bind to GABA receptors and evoke inhibitory neural responses. So I can make sure that you guys have these down in your notes. I just want you to know Okay. So, this is a cell body of a neuron. Okay, if I'm a GABA receptor, that's a chloride channel, okay? Chloride ion channel. So if GABA binds to the chloride ion channel, it's going to open the chloride ion channel. That's going to cause more negative charges to come in the cell because chloride's driving forces into the cell. That's going to cause an IPSP, and that's going to make it less likely that this neuron is going to reach threshold, right? So GABA is an inhibitory neurotransmitter. Glutamate is also important to talk about because it's the most abundant neurotransmitter in the central nervous system. Glutamate which is also a derivative of an amino acid. Glutamate activates cation channels. So you've got two types of channels that it activates. There's one called the AMPA. So glutamate will bind to the AMPA receptor, and the AMPA receptor is actually a sodium and potassium channel, just like the nicotinic acetylcholine receptor. Remember, the driving force for sodium is way higher, so you get if you have sodium rushing into the cell, are you going to have an IPSP from this or an EPSP? EPSP, right? So this one is excitatory. But glutamate also is an agonist for the NMDA receptor. And this is an example of a calcium-mediated postsynaptic potential. So the NMDA receptor is a calcium channel. So if I'm glutamate and I bind to my receptors, not only am I causing depolarization due to calcium rushing in through the NMDA receptor, but I'm also causing the AMPA receptor to open up a conductance to sodium. So you're going to get depolarization. Now, some neurons are going to have AMPA receptors, some neurons are going to have NMDAs, some neurons are going to have both, and so that the response from glutamate is going to depend on which receptors you have. Either way, glutamate is an excitatory neurotransmitter, right? Because it's going to lead to EPSPs. The fourth class of neurotransmitters, neuromodulators, are the opioids, right? So morphine falls into this category, although you don't make morphine, you make chemicals that are very similar to morphine, endorphins, right? Endorphins are part of this class. There's one called cholecystokinin, neuropeptide Y, PYY. These are all peptides. What a peptide is it's a short protein. It's just a protein that's like, 15 to 20 amino acids long as opposed to hundreds. And these are important chemical mediators in your entire body. I mean, CCK actually stimulates enzyme secretion from the pancreas for digestion, but it's also a neurotransmitter. So some of these peptides are used in more than one fashion. Opioids are a perfect example of neuromodulators, meaning that they're pain relievers, right? Most of us know that they're pain relievers, and they act via neuromodulation. So they change synaptic strength. They're going to change gene expression so that you're less responsive to pain signals. The others, there are some gases, so nitric oxide and carbon monoxide can act as neurotransmitters or neuromodulators. We'll talk about nitric oxide a little bit when we talk about reproduction, because we're going to talk about the mechanism of Viagra. ATP itself and other purines. So ATP, you guys know, is used for energy, but it's also used as a neurotransmitter. So just like calcium is used for a bunch of different things, so is ATP. It's not just used for energy, it's used in this case to bind receptors and cause neurological changes. So putting it all together, a neurotransmitter activates its receptor on the postsynaptic neuron, And you have to ask yourself, what kind of signal results, right? What is it called? What is that signal called? If a neurotransmitter binds to its receptors and there's some sort of biological change, what is the biological change called? It starts with a P and it ends in SP. What does that acronym stand for? What's a PSP? Post-synaptic potential, right? Is it all or nothing, or is it graded? It's graded, right? If you get a lot of the signal, it's going to be more pronounced. If it's got a little signal, it's still going to give you a little bit of electrical change, right? So the kind of signal that results is called a post-synaptic potential, right? And if it's an excitatory neurotransmitter, it's going to lead to an EPSP. If it's an inhibitory neurotransmitter like GABA, it's going to lead to an IPSP. And the summation of those responses is going to determine whether you reach the threshold potential. So ask yourself, what is summation? That's pretty simple. Like it would have to be within the same period of time, right? Those signals aren't going to be summated. If I stimulate like the GABA receptor 1 minute, and then I let a minute go by, and then I stimulate with glutamate, those aren't going to be summated, right? They have to happen very close in time, right, to be summated. So summation is just the combination, the combinatory effect, the additive effect, or subtractive effect, depending on whether they're EPSPs or IPSPs. And the summation of those particular EPSPs and IPSPs that are in a very similar point in time are going to determine whether you reach the threshold potential. If threshold is surpassed, then what occurs on the axon? This is a little phrase, but it asks so much, right? You should be able to give me the 7 basic steps of an action potential and what proteins are responsible for those 7 steps, and the mechanism of an action potential. Which channels are involved, but not just which channels are involved, but what aspects of the channel cause certain characteristics of the action potential, right? Why does the action potential all of a sudden stop going up? Spontaneous inactivation and the delayed potassium channels open, right? So those are examples of what I mean. We talked a lot about myelin today. If you have a myelinated axon, what occurs, right? What occurs that would be different than a totally unmyelinated axon? Why is that important? Okay, now the action potential reaches the axon terminal, the voltage-gated calcium channels open. What is the result of sudden increase in calcium in the axon terminal? What happens, right? Think V snares, T snares, neurotransmitter-filled vesicle, right? And then finally, I just leave you with this image, and then we'll take a little break and do this, this case study. But this image reminds us that our brains are very complicated, as is our peripheral nervous system. And you have 2 phenomena that are called convergence and divergence. So first of all, as we mentioned this morning, you can have hundreds, probably thousands of neurons that could converge on one. Meaning that there might be all this entire class of neurotransmitters and neuromodulators you could have being released on one neuron at any given time. Or certain combinations, depending on which ones are active. So imagine if this is a cholinergic and this is a dopaminergic. And if this one's super active, I'm going to get more of a cholinergic-like response, right? But if they're both active, that's going to cause some changes that are unique to both of those neurotransmitters, versus if I just have my dopaminergic active and my cholinergic off. So the way that these are firing are incredibly complex. This still would be called a synapse, but you're going to have many presynaptic neurons and one postsynaptic. On the flip side, you can have one neuron, and different types of neurons actually can have multiple axon terminals. So even though this one neuron is going to send one action potential, and that action potential is going to spread to all of the axon terminals, and this neuron is what? Is it going to release many neurotransmitters or a single one? A single one. Each type of neuron can only release one type of neurotransmitter. But it can release it on a bunch of different postsynaptic neurons. And that's called divergence, right? reminds us, and again, like I don't pride myself as being an anatomist at all, so neuroanatomy like makes my head spin. But think of the complexity of the nervous system just based on this kind of level of anatomy, and then based on how many receptors you have, and neuromodulation, and how you can change the expression of those proteins, right? And that learning is an example of this neuromodulation. That if you hit those synapses and you fire those action potentials on that particular subject, right, that's going to strengthen those synapses. So that's a perfect example is long-term potentiation and learning. What else was I going to tell you about that?